Namaskar. Today we are going to study about the positive regulators and negative regulators of cell cycle. Cyclin dependent kinase, the positive regulators, P53 retinoblastoma, and the Cdk inhibitory proteins, the negative regulators. So welcome to PK's classes. So before that, let us just recall what is cell cycle. Cell cycle consists of two phases, interphase and the mitosis phase. Interphase is the period for preparation of cell division, and mitosis is the actual period of cell division. And interphase again consists of G1, S and G2, three phases. Gs for synthesis, DNA synthesis, where chromosome duplication occurs. G1 is pre-DNA synthesis, G2 is post-DNA synthesis. In the G1 phase there is duplication of the organelles, and in the G2 there is cytoplasmic growth, and this cell is preparing itself for mitosis. The mitosis consists of four phases: prophase, metaphase, anaphase and telophase. So one cell undergoes this phase G1, then S, then G2, then finally mitosis, and one cell will become two cells. So one parent cell becomes now two daughter cells, two daughter cells from one parent cell. So then these daughter cells again can undergo the cell cycle. So that's why we call it as a cycle. So G1, S, G2, M, then again G1, S, G2, M. And before entering the G1, this cell remains in the resting stage. The cell remains in the resting stage until it again enters into the cell cycle. The permanent cells like skeletal muscles, the cardiac muscles, they are permanently arrested in this G0 resting stage. So before the DNA synthesis there is a checkpoint 1, and before mitosis there is another checkpoint 2. So the positive regulators are cyclin and cyclin dependent kinases, Cdk. The negative regulators are P53 retinoblastoma gene and Cdk inhibitory protein, CIP. So the positive regulators help the cell to enter into the cell cycle, into the G1, and the different cyclins and cyclin dependases, their function to help the cell to enter from G1 to S, then S to G2, G2 to M. Whereas the negative regulators, they act at the checkpoint 1 and checkpoint 2, and they if there is not proper DNA to be synthesized, then at checkpoint 1 it will arrest the cell cycle, and again it will check before mitosis, and if the cells going to be formed are not proper, then at checkpoint 2 the cell cycle is arrested. So the cyclin can be of two types mainly: G1S cyclin and G2M cyclin. So G1S cyclin, three cyclins are there: Cyclin A, Cyclin D and Cyclin E. And G2M cyclin, under it Cyclin B is there. Similarly, the cyclin dependent kinases are Cdk1, Cdk2, Cdk4 and Cdk6. Cdk inhibitory proteins, there are different Cdk inhibitory proteins or CIP are there, and these Cdk inhibitory proteins, example is P27, P21, P16. So Cyclin D, which acts on the G1 phase, and the Cdk is Cyclin D forms complex with Cdk 4 or 6. So cyclin functions by binding with its specific Cdk. So Cdk 4 or Cdk 6 binds to Cyclin D, and the Cdk inhibitor protein which can bind both to cyclin and Cdk to inhibit their action is P16. Then in the G1S cyclin, which helps in moving the cell from G1 phase to S phase, that cyclin is Cyclin E. It binds to Cdk2, and the Cdk inhibitor protein is P21. Then S cyclin is Cyclin A. Specific Cdk is Cdk2. Cyclin A Cdk2 complex can progress the cell through the S phase, and P27 binds to both Cyclin A and Cdk2 to inhibit their action in the S phase. Then G2 cyclin is Cyclin A again, but the Cdk is Cdk1. So Cyclin A Cdk1 complex acts in G2 phase, and Cdk inhibitor protein P27 also inhibits this. So Cyclin A Cdk1 or 2 complex is inhibited by P27. Then Cyclin B and Cdk1 complex help the cell to progress from G2 to mitosis phase, and the Cdk inhibitor protein here is P21. So if you see this in the cell cycle, you can see here in the G1 Cyclin D Cdk4 or 6 complex is there. It is inhibited by P16. Then from G1 to S, Cyclin E Cdk2 complex inhibited by P21. In the S phase, Cyclin A Cdk2 complex inhibited by P27. G2 phase, Cyclin A Cdk1 inhibited by P27, and in G2 to M, Cyclin B Cdk1 inhibited by P21. So the cell division, the stimuli is mitogen, which acts on the mitogen receptor, which is a tyrosine kinase linked receptor. So there will be dimerization, and the RAS-RAF pathway is followed. So RAS GDP is converted to GTP exchange will be there. You can follow our RAS-RAF pathway, tyrosine kinase linked receptors. Then that RAS GTP then can transfer the phosphate to RAF, then RAF to make to MAP kinase, and this MAP kinase then enters into the nucleus. So this receptor is located on the cell membrane, and then it enters. So here RAS is there, RAS GDP. So there will be GDP exchange. Then that will activate the RAS GTP will activate the RAF. RAF will activate make will activate MAP kinase. MAP kinase now enters into the nucleus, and it will help in transcription of the immediate early genes like MYC, and this MYC will then induce the transcription of delayed response genes like cyclin and cyclin dependent kinases, which help in cell division. So in this way, mitogen acts as a stimuli for cell division. And when similarly here there is another transcription factor E2F, which is activated by Cyclin D Cdk4 or 6 complex. So this E2F is in inactive form when it binds to the retinoblastoma. So retinoblastoma inactivates E2F. That's why we call this retinoblastoma RB as a negative regulator. RB, retinoblastoma gene is a so from retinoblastoma gene there will be production of retinoblastoma protein, and that retinoblastoma protein acts as a negative regulator. That's why we call this retinoblastoma gene as a negative regulator. And the Cyclin D and CDK4/6, so this MIC can produce cyclin, and Cyclin D, CDK4 or CDK6 complex can activate E2F by detaching it from retinoblastoma, and this E2F acts as a regulator of S phase gene transcription. It causes S phase gene transcription, so there will be transcription of G1 S cyclin and S cyclin. As we have discussed here, MIC helps in transcription of cyclin and cyclin dependent kinases. So here Cyclin D and CDK4/6 complex can activate E2F, then E2F will cause the transcription of again the cyclins, Cyclin E and Cyclin A. So the Cyclin E is a G1S cyclin which will make this G1S and CDK active, CDK complex active. As we have discussed here, Cyclin E and CDK2, Cyclin E and CDK2, Cyclin A and CDK2. So these complexes will be activated, and accordingly the cell cycle will progress. So S phase gene transcription helps in activation of G1S cyclin and S cyclin. G1S cyclin like Cyclin E, S cyclin like Cyclin A. G1S cyclin will activate G1S CDK complex, which will activate again Cyclin D CDK4, and S cyclin will activate the this S CDK complex, which will help in DNA synthesis. So in this S phase it will act. E2F helps in your S phase of cell cycle, and it is inhibited by retinoblastoma, so retinoblastoma acts as a negative regulator. Then another negative regulator is P53, which with MDM2 forms a complex and becomes inactive. So P53 with MDM2 is inactive, and when it will be free from MDM2, it becomes active. So when there is excessive MIC, so usually MIC induces the cell division, but when there is excessive MIC, that will activate another one, this ARF, which will form the complex with MDM2 and will activate P53. So you can see here P53 MDM2 complex. ARF forms complex with MDM2 and releases P53. That is, ARF phosphorylates this complex to release P53. So this one now active. This occurs when there is excessive MIC. Similarly, this P53 acts as a negative regulator when there is DNA damage. DNA damage also is a stimuli to activation of P53, and DNA damage causes activation of ATM kinase and ATR kinase. ATM, the full form is ataxia telangiectasia mutated. ATR is ATM and RAD3 related. These are the members of phosphatidylinositol 3 kinase family group of enzymes. Then they will recruit and activate CHK1 kinase and CHK2 kinase, which will phosphorylate this P53 MDM2 complex to release the active P53. So this P53 can be activated by, which is usually in inactive form when it is with complex with MDM2, and this becomes active when there is either excessive MIC or when there is DNA damage. So this P53, how it acts, it binds to the regulatory region of P21 gene, which is a CDK inhibitory protein, and there will be transcription of P21 gene to P21 mRNA. Then there will be translation to P21 protein, and P21 protein can inactivate G1S CDK as well as G2M CDK. So it will inactivate the Cyclin E and the CDK2 complex, and similarly it will inactivate G2M Cdk, that is cyclin B and Cdk1 complex. So by inactivating the cyclin E Cdk2 complex, it arrests the cell cycle at checkpoint 1, and by inactivating the cyclin B Cdk1 complex, it arrests the cell cycle at checkpoint 2. You can see, so cyclin E Cdk2 complex at checkpoint 1, it will be inhibited, and this is by inhibiting cyclin B Cdk1. So P21 inhibits it. It is checkpoint 2. So it is checkpoint 2, checkpoint 2, and this is checkpoint 1. So by P21, so checkpoint 1. So P21, that means the negative regulator P53 arrests the cell cycle at checkpoint 1 as well as checkpoint 2 through P21. So P53 binds to the regulatory region of P21 gene and produces P21 protein and inactivate cyclin E Cdk2 complex, cyclin B Cdk1 complex, and accordingly arrest the cell cycle at both checkpoint 1 and checkpoint 2. So this is all about this cyclin dependent kinases and Cdk inhibitor proteins. Thank you.